everyone. In our ongoing series of biotechnology, today I, Dr. Charu Dogra Rawat, Associate Professor at Ramjas College, University of Delhi, will be discussing about genetic gene editing tools. The learning objectives of this session would be to understand about the gene editing. I'll be giving an overview and talking about the mechanism, the outcome, and the timeline of gene editing technologies. I would also be the objective would also be to understand the homologous recombination as a gene editing tool, which is historically the first kind of gene editing tool that was utilized. Now, what do you understand by gene editing? Gene editing or genome editing is a method for making specific changes to the DNA of a cell or an organism. It can be used to add, remove, or alter DNA in the genome. As we will see, it is based on engineered or bacterial nucleases. The main idea is to modify the DNA for our own purpose. Now, biotechnology is basically about the biology and developing it into the technology. So, the nucleases are present in the cell, and these nucleases cause the DNA to be broken, and the homologous recombination causes the DNA to be repaired, which create new forms of DNA or basically causes the changes in the DNA or alterations in the DNA. Now, to utilize this alteration or modification in the DNA for our own purpose and benefit, this genome editing technology is developed and various tools are utilized, such as the engineered or bacterial nucleases for the gene or genome editing. Now, as I said, historically, homologous recombination was the earliest method used to edit the genomes in the living cells. Now, it is a natural phenomena that occurs, and it is utilized for the repair of the DNA whenever there is some kind of an error or to create certain variations. The homologous recombination occurs, and this method was actually utilized in biotechnology to edit the genomes as per our interest. It involves the exchange or what is called as the recombination of genetic information between two similar homologous strands of DNA. Now, the important part is the homologous, which is basically the two similar strands of DNA, and it involves the exchange or recombination. So, either the two strands, when they get exchanged, the recombination word, we also started with the biotechnology series with recombinant DNA technology, where two DNA coming from two different sources are recombined together. So, in here also, because two different DNA strands, they come together, it is termed as the recombination as well, and also the exchange of DNA. So, that is primarily the basis of the homologous recombination. How do we utilize it in the laboratory or in biotechnology processes? The isolated undamaged DNA fragments bearing genome sequences similar to the portion of the targeted genome are injected into individual cells or taken up by the cells using special chemicals. So, basically, if it is the eukaryotic cells, the transfection is done, or in prokaryotic cells, even the transformation is done, where the isolated and undamaged DNA fragments are introduced into these cells. They can be injected into these individual cells, or they can be transformed or transfected utilizing different chemicals. So, when this DNA get introduced into the cell, then the homologous recombination can occur. The important part is again that the portion of the targeted genome must have certain sequences similar to the targeted genome. That is when the homologous recombination will take place. Now, once inside a cell, these DNA fragments can then recombine with the cell's DNA to realize the targeted gene addition, replacement, or inactivation. Now, one word which has been utilized in these 2-3 slides is what is called as the targeted gene addition. Now, because there has to be some homology between the introduced DNA and the genome where we want to make a change, it is termed as gene targeting as well. And in this gene targeting, the addition, replacement, or inactivation can take place by when this exogenous DNA get recombined with the cell DNA. So, for this recombination to occur, homology or homologous chromosomes or homologous portions of the DNA needs to be there. So this is a very schematic diagram, a very generalized diagram which tells us that this is the genomic DNA and this is the targeting vector. So this is basically the vector which is carrying our gene of insert. We have elaboratively talked about the recombinant DNA technology, that how such vectors can be created that carry our gene of inserts. They generally do carry a selectable marker as well, and as you can see, it is flanked by the homologous sequences. Now these homologous sequences have certain homology to the genomic DNA, and as you can see here, they have been introduced or integrated by the homologous recombination, and this insertion, then in the second round the accession of the selectable marker can take place and then the DNA is inserted. So in this particular case there is integration that is happening or insertion that has occurred, but that is how basically the homologous recombination is there. This is an overview of that. So this can be insertion, there can be integration, there can be replacement, or there can be inactivation or deletion as well. So this is what we require to do. This is the sense of gene editing or genome editing, and which has been accomplished by this homologous recombination. So this was the earliest method how it used to be done. The homologous region flanking the inserts are basically introduced into the cells, and there they can homologously recombine, and therefore the additions, replacement or deletions can occur. This HR gene editing is extremely inefficient in most cell types, because the introduction into the DNA as well as it is not easy, or it is not applicable for all the different cell types, particularly mammalian cell types as well. Then this HR is also inaccurate and has a high rate of error when the injected DNA fragments insert into an unintended part of the genome, causing what is called as off-target edits. Now in any gene editing process, as we will see subsequently, this off-target edits is a problematic situation, because we have homologous sequences flanking the genes of insert, and they show the homology to the genome. They may show homology to different parts of the genomes and not just one specific part where we want to make a change. So these off-target edits, or sometimes the random insertion can also take place, or deletions can also take place. So these off-target edits are really problematic, and therefore this homologous recombination as such, it is not an efficient process, because the entry into the cell types is really difficult, and secondly it is also inaccurate and has a high rate of error, which makes and urged or compelled the scientists to discover many other gene editing tools, very efficient gene editing tools, and it has still been discovered. An important aspect was the generation of nuclease-induced double-stranded breaks that leads to the stimulation of highly efficient recombination mechanism of cellular DNA. Now, because the natural homologous recombination process on which we were relying in the earlier gene editing mechanisms, it was observed that the nucleus-induced double-stranded breaks or DSBs, it leads to the stimulation of this recombination mechanism of cellular DNA. This nucleus-induced DNA DSBs can then be repaired by one of the two major mechanisms that occur in almost all cell types and organisms, and these two mechanisms are the homology directed repair and non-homologous end joining, resulting in targeted integration or gene disruptions respectively. So creation of these DSBs was used as a pre-step before, it's the first step for the gene editing mechanism to take place, and it really increased the efficiency if we left it for just the natural homologous recombination that was occurring. So many nucleases were engineered, or biological nucleases were used to create these DSBs. We will talk in detail about these tools, how they create these DSBs, and we again talk in detail about the mechanism, the two mechanisms by which the DNA insertions or additions or deletions or gene editing can take place, the HDR and the NHEZ, and we also discuss about what are the different kinds of outcomes of this gene editing that takes place is. So looking at the two mechanisms, the first mechanism is the homologous directed repeat. So we have talked about the homologous recombination, where there is a homologous part that combines with the targeted genome, that basically interacts with the targeted genome, and then there is an exchange of the strand that occurs, resulting in the gene editing. Now this HDR is a repair method. It is used as a repair method when there are double-stranded DNA breaks that can occur. So this is the basic method that occurs for HDR. So whenever a double-stranded break is created here, then there are proteins that get associated with this double-stranded end breaks. There is 5' 3' resection that occurs. So as you can see, there are certain nucleases that associate here. Variety of nucleases. We are talking about the overview, and in any case these homology directed repeats as well as the non-homologous end joining methods have been talked in detail in our molecular biology series, where we talked about the DNA repair mechanism. But I just review it here because it is more relevant to what we are discussing and for the understanding of how the gene editing is taking place. So when the nucleases, we'll talk about the different types of nucleases that create these double-stranded breaks. But when these double-stranded breaks are created, then how these are repaired and how these repairs causes the gene editing to be done is what I am showing here. So in HDR, various type of proteins, as you can see, they will come and lie at the end of the wherever the double-stranded breaks are there. They will identify these breaks. There is a 5'. There are certain nucleases which will chop off the 5', 3' resection. So they basically chop off in this direction, and you will have a single-stranded overhangs, as you can see here. This recruits then the replication protein A, such as the RPA molecules, and these RPA molecules basically stabilize these ends and causes the recruitment of other factors and proteins such as RAD proteins. Now these RAD proteins cause the identifying of and searching of the homology fragments in the genomes. So this is what has been done, and then they basically search for the homologous sequences in the chromatids. This is basically happening in the synthesis phase of the cell cycle, and in the G2 phase of the cell cycle, when there are chromatids that are homologous chromatids that are joined together. So they search for the homologous sequences in the sister chromatids, and then these are the two mechanisms by which these can be resolved. So there are these kind of D-shaped loops that are formed, and they can be resolved by either non-crossing over, where the synthesis of the strands takes place, and the crossing over bridges, where basically this is called as the Holiday junction, named after its discoverer, and this is resolved by the synthesis again of the strands based upon the homologous template. So these are the regions which are homologous in between the targeted one and in between the sister chromatids, and then the synthesis of the strands, the complementary strands take place, and ultimately ligases come and seal this nicks which is there. So this is the homology directed repeats. So there has to be a break, a double-stranded break, and then the set of proteins that stabilize that breaks causes the 5', 3' resection, causes the proteins to again come and join, causes the different proteins assembly at these breaks, and where there are single-stranded 3' overhangs. These proteins stabilize them, and then they further recruit certain proteins, which helps in the identification of the homology templates, and then they are extended by the polymerases, yet unknown polymerases, and these polymerases extend it, and then the ligases come and seal this off, and therefore the new, based upon the homologous sequences, the sequences are formed here. So the targeted genome sequences are basically integrated into the genomes out here, as you can see, and these are the portions which are basically either inserted or deleted or whatever. The further resolution will take place where, in this case, for example, the replacement has occurred. There can be integration as well, or there can be disruption as well, or deletion as well that can occur. This is a homology directed repeat that is occurring. This is a repair mechanism, a natural repair mechanism which is present in all cell types. But when we engineer nucleases to create double-stranded breaks, then when they are repaired, they basically causes the gene editing to be done, because we are creating the double-stranded breaks at our desired positions. We are probably introducing the target DNA, we are introducing the desired DNA showing homology to the targeted DNA, and thus these kind of crossing over or non-crossing over repairs can occur inside the cell, causing the gene editing to occur. The second mechanism is the non-homologous end joining method, which involves the following steps. The first is the DNA end recognition and assembly and stabilization of the NHEJ complex at the DNA double strand break. Then the bridging of the DNA ends and promotion of the end stability. The third step is the DNA end processing. And the fourth step is the ligation of the broken ends and dissolution of the NHEJ complex. Now let's look at these steps a little bit in detail. So the first step is the DNA end recognition and stabilization of the core NHEJ factors. So sometimes when there are double-stranded breaks, then those double-stranded breaks are identified by a series of protein. They are recognized by a series of proteins, and these proteins basically tend to accumulate at these ends, and they further introduce or they further recruit various NHEJ machinery in which there are a role of nucleases, there are ligases, and there are other protein molecules which are present here. So as you can see on the ends, these NHEJ factors, they are recruited. So this is basically the first step where the DNA recognition is there, the DNA stabilization is there by these proteins, and then further recruitment of the NHEJ core factors that occurs. Then after that, the DNA end processing occurs, and in this DNA processing, as you can see, the two different methods are shown here. One where there is a kind of complex DNA repair that has to occur, and one is more of a simpler one which recruits various kinds of proteins including artemies, etc., and then further nucleases are also added here, and all these complexes basically process the ends for the ligation or ligase to act upon. So as you can see, the various end processing has occurred and a series of number of factors and proteins are involved in it. So these proteins, this is basically a simple pathway related to some factor as mentioned here, and these are related to when it is a complex kind of a thing, and various others such as ATM proteins and Artemis proteins are used here. So there are a number of proteins that are involved that process the end and stabilize them and make them ready for the ligation to occur. The last step is the ligation of the DNA ends and dissolution of the NHEJ complex. So there is a phosphorylation, dephosphorylation that occurs. So these molecules are the kinases. So these kinases that has been introduced up here, so these are the PCA kinases which are there. So these kinases causes the dephosphorylation or causes the phosphorylation of various proteins that brings on the conformational changes as well as activate them. They activate the ligases as well. So in fact at these points only, these kinases when they are activated, they cause the DNA end processing to be there, further recruiting certain other protein molecules, and then they activate the ligases as well. These ligases call the ends to be joined, and these then the NHEJ complex, because of the dephosphorylation of the components, it disassemble and the NHEJ complex then releases, giving rise to the end joined or the repaired DNA. And in this repairing basically, because our own nucleases have created, our engineered nucleases or specifically used nucleases have caused these double-stranded breaks. So there basically the gene editing can occur at this point. So these are the two major repair mechanisms that are utilized. When the double-stranded DNA breaks are there, we'll talk about the different nucleases subsequently, which causes the ends to be, or the DSBs to be repaired, leading to various kind of gene editing methods. So what are the kind of genome editing outcomes that is occurring? So as you can see, there can be knockouts, there can be replacements, there can be gene corrections, there can be inversions that is occurring, there can be integrations, there can be basically gene deletions or the gene correction. So what is happening here, that when a double-stranded break is created inside a gene and that the function of the gene is abrogated, it is removed after the repair mechanism. When the gene is repaired in such a manner that its function is removed, then we create what is called as a gene knockout. Or the entire gene is removed here. So the function is removed. The entire gene is removed here. So you can see the double-stranded end breaks is caused at the end of the genes. So this is basically knockout or knock-in. There are different methods which are there, and the entire gene can be removed. Again, when this part is removed and somehow it is linked or repaired, when the repair happens, then they are done it in the inserted manner. Then there is called as the gene insertion method. In this case, there is basically what is happening is there was a particular gene, and when a double-stranded, so this is a targeted thing. This is happening when the double-stranded breaks are created in the gene DNA. This is gene integration. When we introduced our gene of interest in a targeted, in a vector and targeted it to a particular site, then the gene insertions can occur. Or there is a mutated copy of the gene in the genomic DNA, and we introduce a correct copy. This is the essence of what is the gene called as the gene therapy. So the diseased gene or the mutated gene, we cause the double strand breaks in that gene, and during the repair mechanism, for example by the homology mechanism, directed repeat, we cause this particular portion to be introduced here, and in this way basically the gene is corrected. So this is basically the essence of gene therapy, as I said, and this is termed as gene correction. So there are many different gene editing outcomes that can occur. There can be insertions, there can be deletions, there can be reactivations, there can be inversions, there can be gene in, there can be gene knockouts or gene knock-ins. So different kind of genome editing outcomes are there, which is dependent upon the basic mechanism that we have discussed. There are many gene editing tools. The major gene editing tools are that we are going to discuss is the homing endonucleases. There are what are called as zinc finger nucleases. So they contain two parts. One is the zinc finger protein that causes the specificity. These are the DNA binding domains. And an endonuclease, FOX1 endonucleases, and they basically attach in this dimer portion to the double-stranded breaks and cause the repair to occur. So, and they create the double-stranded breaks, and subsequently the repair can occur. So they bind specifically to the site recognized by these zinc finger proteins which are there. We'll discuss in detail about them. Then there are what are called as talons. And these talons again are basically just like ZFNs, but they contain long, these kind of nucleotides basically. They are the talons, how they are going to cut. And then the, newest, not the newest, but the widely accepted and most efficient method, the CRISPR-Cas9 system. So in this CRISPR-Cas9 system, we have got a Cas9 protein. And then these CRISPR molecules basically attach to the Cas9 proteins, and they recognize certain specific sites on the DNA, and then they create the nicks and they create the double-stranded breaks or the nicks that can then be repaired and cause the genome editing to be occur. So now this is the most efficient method that is utilized. So we talk in detail about these genome editing tools in our subsequent sections. Thank you.